let's get started. It is 1:06 p.m. I guess. Uh, Whatever.
Day
Anyway,
17.
today we're gonna learn about some DNA replication. I hope you guys did some kind of reading, so you know what we're learning generally, because this is really big. Lots of stuff to understand here today. Let's get started. So before we get into all the details, we are going to look at a few different models of DNA replication. So, uh, yeah, let's get started. So we're gonna cover like when replication happens, three different models. Not all of them are correct. One experiment that basically proved which one is correct, and then we'll kind of get started on the details. So, I think we learned the cell cycle many times. Yeah, we did for sure. And we know that there's a bunch of different phases to the cell cycle. DNA replication happens in S phase, right? This is not new information. I hope everybody knows this. Or if you didn't know already, yeah, it happens in S phase. G1, as you can see here, it's when all the cellular contents, excluding the chromosomes, are duplicated. And then in G2, the cell double checks the chromosomes, making any repairs. And then you guys know what happens in mitosis. And that's what we basically studied in previous grades. We studied mitosis. Okay, moving on. So there are three different possible models of DNA replication, and not all of them are correct. If you may know which one is correct already, but we're gonna basically look at the three of them and how they differ. It's my dog. Uh, just ignore her for now. Okay, so starting at the top, there is one parent double helix. If we look at the first model, the conservative model, basically it's conservative because the parent DNA is conserved. So the parent DNA is not changed, and we say that an entirely the parental double helix remains intact and an all new copy is made. So that's the conservative model. Oh, I think I went forward. Or I guess we can move forward to this. No, stay here. In the semi-conservative model, we say that the two strands of the parental molecule separate, and then after that, each strand of the parental molecule functions the template for the daughter strand or complementary strand. And then in the dispersive model, we say that basically the parent DNA molecule is dispersed. So each strand of both daughter molecules contains a mixture of old and newly synthesized parts. So it's basically like like completely conserved and versus like not conserved really. So that's the three models. And yeah, this is basically just overview, a little bit easier to see. Stay here for a couple seconds. Right, move on. So in 1958, Matthew Meselson and Franklin Stahl, they performed an experiment to determine which of the previously the three models we looked at was true. So the way that they went about doing this is actually it's really smart. It's actually very clever. So we know that DNA contains nitrogenous bases or nitrogen atoms. So what they did was they grew E. coli cells in heavy nitrogen. So when these cells are centrifuged, the DNA will show up heavy or heavier than normal. So I don't know how they went about doing this, but it's basically they created a generation zero where the DNA is made entirely of this heavy nitrogen. And there's a centrifugation method that basically creates a density gradient. So what they do is they use like a pigment. I don't know what they use, but somehow after the centrifugation, they can create basically like these bands that show different densities. So this is really important because let's say that you have like a heavier cell, then it will show up more dense in the after the centrifugation. So, any questions? All right, move on. So, step two, you take these uh, E. coli that have been created in heavy nitrogen, and then you let them replicate once in a medium with light nitrogen. So they are only, the new cells are basically the heavy nitrogen E. coli are only allowed one round of replication, and then they're centrifuged again. So if we say that there's a conservative model and one round of replication has happened in heavy nitrogen and then another one in light nitrogen, could you guys maybe hypothesize what the conservative model, basically what the DNA might look like if it was a conservative model?
Do we raise our hand? How does this work?
I don't know, just
I guess you can just talk.
yeah, just talk.
It would just, you'd get two strands. So one strand would be both. It would still be the heavy, and then you get another one with two of the blue ones.
So like in
Okay.
the centrifuge,
Yeah,
like
I would agree with that.
the pink one would show up first, or all at once, and then the blue one would show up, right?
So we'll get one blue one
And then one pink.
and one pink one, like this. And this would be, oh, whatever, I'll just keep that. And let's change to black. So the blue one would be light, and the black one, or sorry, the red one would be heavy. Does everybody agree?
Yep.
Mm-hmm.
Wait, so in
Cool.
the centrifuge too, it would be all red and then all blue, right?
Yeah, so if we had that in a centrifuge, oh, maybe I should switch to red. So we would have like, uh, what's going on? Right. So I don't know. We'd have like a red zone, and then one second.
Mic off. Mic on.
Okay, so yeah, we'd have a red zone, and then we'd have a blue zone, a little bit higher up. Okay. Right.
Mm-hmm.
So I know this is slow, so we'll do everything for this time, and then the next one's like we can go a little bit faster. So semi-conservative. What do you guys think it would look like?
Mic
Purple.
feedback off.
Purple?
Mic feedback on.
If you mix them.
Oh yeah. Okay. I mean, I was more expecting like, oh, that's kind of bad. I was more expecting like, okay, let's have like one blue
One strand blue, one strand red.
and then one strand red.
Purple.
Oh, it's a different blue, but I mean, you guys get the idea. And then like this times two, right?
And there'd only be one band because they're the same density now.
Exactly. That's what's really important here. And purple or whatever, one band. Okay. And dispersive. What do you guys think?
Same thing, but like looks a little different.
So
I guess it'd be pretty similar because it's still the same amount of blue and red.
exactly. So could you really tell right now at this stage? Could you like tell between? I mean, it's kind of hard to draw, but could you really tell between a semi-conservative model at this point and a dispersive model?
No.
This
I don't
drawing's
think so.
this drawing's kind of bad, but I mean, you guys get the idea. So no, at this point you can't tell. So this is what they knew this, and here all that they could really go off is this is two bands, this is one band, and this is also one band,
Mm-hmm.
right? So what do you think they saw in real life?
The
I
same
don't know.
thing.
So what they saw in real life is one band. They saw one band, so immediately they knew that the conservative model is ruled out. But they can't tell yet between the semi-conservative and dispersive model, so what do? They allow one more round of replication, and then they centrifuge one more time. Does that make sense? If it doesn't make sense yet, it'll make sense soon. So, assuming that, the previous step we do it, and then we allow one more round of replication as is. Okay? Let's go through what the DNA would look like. Conservative model, what do you guys think it would look like?
It'd be two red and two blue.
Two red,
If you're taking.
two blue.
If you.
But you see, so let's say we had the heavies. Okay, so we had one heavy from the original.
Mm-hmm.
So the previous step, this is what it looked like. right?
Yeah.
But you still only have light nitrogen.
Would you have 2 mixed and then 2 blue?
But based on the conservative model, this one is
Oh, it'd be
preserved.
red, blue, blue, blue.
There we
Oh,
go.
3 blue, 1 red.
There we go.
Sorry.
3 blue, 1 red. That is the answer. So it goes like this.
Oh, I see, I see.
Yeah. And then let me draw the red one. Yeah. Does it make sense?
Mhm.
Yep.
And if we centrifuge this, let's get the red. The red, there would continue to be like a red band, and there'd be, I guess, just like maybe a thicker blue band. But yeah, but it's still 2 bands, correct?
Yep.
Yeah. Alright, so now let's go through the semi-conservative model again. Assuming that we have a semi-conservative model, if we had 2 red blues last time, so let me go like this. Yeah, pardon my drawing, but hope you guys get the idea. So we had 2 of these last time. What happens this time?
2 blue, 2 mixed.
Mhm.
2 blue, 2 mixed. So and then here, it's taking a while, but it's worth it. Okay, so this is what we would see, right?
Mhm.
Do you guys agree? So
Mhm.
basically on this side, like the blue strand is the template, and on this side the red strand is the template. Same thing on this side. So when we centrifuge it, does our purple band go anywhere?
No, it's still there.
Yeah,
Uh,
purple band still stays.
with the
And
blue band.
now there's a blue band. Oh, good. Okay, finally, the dispersive model, which looked like this before. What do you guys think happens to the dispersive model this time?
It gets even more mixed.
Yeah, it gets even more mixed actually. So before, if it was like a 50-50, now it's like a, I don't know if I know my probability, but I'll just say it's like a 75-25. So I'll just like a 25 red and like a 75 blue, but then there's 4 of them. Do you guys agree?
Yeah.
So if we had something like that, where would the band go?
So it'll just be one band, but
Yep, one band,
It
but
might
like,
be a little,
higher.
a little higher since
there
it's
we go.
less dense.
Yeah. And it would be like, since we're using colors here, it would be more blue than red.
Mhm.
Alright, so yeah, that's what it is. So, after a second round of replication, the dispersive model still only has 1 band. And the conservative model, which also has 2 bands, is ruled out because we know from the first step of replication that it can't be the conservative model. So only the semi-conservative model survives like 2 steps of like, or is correct after 2 steps of replication. So that's how they determined that DNA replication follows a semi-conservative model. So now that we know this, we know that whenever you have a single strand of DNA, each strand will be a template for its complementary. So now we can get into a little bit of the details. So if you guys want to go and study this, like I would recommend like just like looking at this slide. It's pretty useful. This is a pretty important experiment. If you guys ever end up having tests, like this will definitely be on it. But yeah, who knows when the test will be at this point. So there's a bunch of summary and stuff. All right, let's move on. So the basic concept of DNA replication, after knowing all this, is that during DNA replication, base pairing enables an existing DNA strand to serve as a template for a new complementary strand. So let's get into the mechanisms. In this PowerPoint, you'll primarily be covering, oops, give me 1 second guys. I think somebody wants to join the server for the lesson. So let me let them in. Okay. Okay. Right, back to this. Alright, so there are basically three main steps to DNA replication: initiation, elongation, and termination. The steps themselves are pretty self-explanatory, but the details that are here are definitely not, so let's go through it. Replication initiation always begins at something called an origin of replication, or shortened to an ORI. And these are special sites on the DNA where replication begins, and depending on if you're looking at a eukaryote or a prokaryote, the conditions to begin DNA replication are a little bit different. For example, for prokaryote DNA, or in this case a plasmid, you only have one origin. So on the plasmid there's one origin of replication. Replication continues until you reach the termination, and then you have two circles. So this is what this diagram is going, so you follow it like this. But for eukaryote DNA, it's a little bit different. There are thousands of origin sites per chromosome, and basically starting at an origin, DNA replication goes outwards to both sides until it reaches another, basically. So once like this reaches this, it continues replication, and maybe they'll meet somewhere around here. In this slide, I believe we'll primarily be covering eukaryote replication, just because the enzymes involved are much simpler than eukaryote replication or human DNA replication even. So but the ORIs we do cover separately here. And here's a little diagram. Cool. And then, yeah, as I said earlier, the replication bubbles elongate as the DNA is replicated, and then they eventually fuse. Okay, so there are many, many different proteins involved in replication initiation. I don't think we'll cover like all the different like factors and conditions to start it, but like let's say right after starting replication, there's a bunch of different proteins that have to come in and start doing some work. So then, do you guys know what helicase, single strand binding proteins, and topoisomerase is yet? You guys haven't learned it yet, right?
No.
No? Okay. Right. So, this is a little diagram. I know it's like there's like tons of different stuff, but let me go over it and paint really quickly. So, here, let's say this is your DNA. So this is like strand 1 and strand 2, like opposite strands. And let's say that they started replication here. So this is the ORI. And replication is continuing in this way. We'll ignore what's happening on this side for now. We're going to talk about three different like proteins right now. So let's say that there's an enzyme called helicase, and like I'll place it over here for now, or I could even just like place it here. I haven't drawn the bases in for sake of simplicity because it's going to take a really long time. So what helicase does is basically helicase breaks the hydrogen bonding between the bases and allows the two strands to separate. But just because they're separated doesn't mean that they want to stay this way. So if this was yarn, two strands of yarn, helicase is basically unwinding them, but single strand binding proteins, they come here, they basically attach to the strand and prevent the two strands from coming back together, if that makes sense. Does it make sense? It's like basically tape that holds them together or holds them apart. And further up the DNA, you need another enzyme called a topoisomerase. Let's say that you guys have two pieces of string, okay? You guys are gonna have to think pretty hard about this. And I have them like this. Let's say that these two ends can't move. If you pull these two ends of string apart from each other, and you continue to pull, what happens to like the string here? There's a lot of
It's
tension that
pressure.
builds up here, right?
Mhm.
It like coils and a lot of tension is built up. So that's basically what's happening as helicase is moving along and single-stranded binding proteins are preventing the DNA from coming back together. There is going to be tension in the DNA here. So how do you relieve this tension? Because if you don't relieve it, then the DNA breaks. And if the DNA breaks, then that's like a really, really big problem, right? So that is where an enzyme called topoisomerase comes in. And basically what it does is um, it relieves the tension in the strand, you know, top topologically, the topoisomerase. The actual mechanism that it goes through to unwind the strands is a little bit more complicated and it's hard to show you, but essentially if you cut and then allow the string to like pass over each other and then you attach it again, you relieve the tension. It's a lot easier to show you in person, but since I'm drawing this on Microsoft Paint, you might have to take my word for it and then go online and look for a video. Um, if you guys really want, I could probably film something tonight and then just create a short video showing it and then I'll post it here tomorrow. But for now, take my word for it. It's pretty cool. Continuing. Alright, so here's like a little written explanation. Helicase disrupts hydrogen bonds between the two strands. So we know this. Good. Single strand binding proteins. That's pretty self-explanatory. And topoisomerases. Yeah, pretty self-explanatory this part. So now that we know how DNA unwinds, we have to start talking about how DNA is built up again. So from these like single strands here, what happens to create the complementary strand? So we'll get into that right now. Basically, or to begin, we have to know what priming is. Priming DNA for replication. So I want you guys to look at this diagram. This blue line is template. So the blue line is originating from the original strand, the parent strand that we uncoil. The pink line are something called primers. Essentially, they're RNA nucleotides that are added to the template strand to allow another enzyme to come and continue. And they're really important for a specific reason. Light pink blobs are called RNA polymerases, and I'll shorten that to RNAP. So whenever I use the term RNAP, it's going to be referring to RNA polymerase. And whenever I refer to primers, primers are the RNA nucleotides that are added by RNAP to the DNA to prime for replication. I know this is a lot of information. If you have any questions, feel free to ask. So we're going to continue. Why do you need to prime DNA for replication? So you need to prime DNA for replication because DNA polymerase, or DNAP, which I'm going to refer to it from now, DNAP can only add bases to DNA if it has an existing 3' end to add to. RNAP is not limited by this. So RNAP can basically plunk down an RNA nucleotide anywhere on this blue strand, but DNAP can only add to an existing 3' end. And this is really, really important and creates a couple challenges in DNA replication. So yeah, here you go. You can read through it. Due to the different abilities of RNAP versus DNAP, RNAP can start a new chain without an existing 3' end. All it needs is a template strand. DNAP can
Is there a
only...
reason?
Is there a reason?
Sorry. Is there a reason why there's DNAP and RNAP?
Oh, instead of just one enzyme that does it all?
Yeah, instead of just RNAP, let's say.
Oh, it's because RNAP adds RNA nucleotides. It can't add DNA nucleotides.
Oh, I see, I see.
Yeah, so eventually what needs to happen is another form of DNAP is gonna come over this and like replace all these ones. So this doesn't stay.
Oh, I see.
Yeah. Anyway, we're gonna continue.
Wait, sorry, Alex, quick question.
Mhm.
You know how for RNA instead of thymine they have uracil?
Mhm.
Does that uracil just bind to the A? Like it's fine? Is that just what happens?
Yeah, it does.
Oh, okay.
But it
Okay.
can't stay. It
Okay.
won't stay. It could. So there's nothing wrong with uracil binding or binding to adenine because it does that in RNA, right? For
Mhm.
eukaryotes, or sorry, prokaryotes. But in eukaryotes, like we don't want this, and um, primers, yeah, primers won't stay. There's no problem with the primer being here to begin DNA polymeration or polymerization, and it's really required, but it will be replaced. And I couldn't tell you why, other than the fact that it is this way, because I didn't really invent the system. Yeah, so
Okay.
honestly, it is this way. If you have any other questions, feel free to ask. And if I don't know the answer, I'll probably try and look it up, or you can ask Miss Law. Okay, so DNAP. In prokaryotes, there are 5 types of DNAP. In eukaryotes, there are over 15 different types, named with Greek letters. And they form this huge, crazy, like transformers, like complex that like travels along the DNA, and there's so many different parts. So we are going to focus on prokaryotes with their DNAP1 and DNAP3. DNAP1 and DNAP3 both polymerize on the DNA strand, but they have distinct functions. So we'll cover that in a second. So right now we just covered priming, and priming is the adding of RNA nucleotides to begin DNAP function. Right now we're going to look at elongation. So elongation is adding to that primer and basically beginning the DNA polymerization process. In order to add to a 3' end, so this is what DNA polymerase is going to be doing, it's going to look at an OH, it's going to bring in this nucleoside triphosphate, it's going to use basically it's going to use the phosphates, 2 of them, as energy, and then add a nucleotide in its place. So the the circle here is a phosphate, the pentagon is a sugar, and then this little piece is the base. Do you guys understand what's happening?
Mhm.
Good.
Yep.
All right, continuing. DNAP3 catalyzes the elongation of DNA molecules by adding nucleotides to the 3' of an existing nucleotide. So what that means is this thingy here is DNAP3. DNAP1 will make its debut in a moment. As each nucleotide is added, the last 2 phosphate groups are hydrolyzed to form pyrophosphate, and pyrophosphate is broken down into 2 phosphates. This is basically just energy. This detail is a little bit less important, but cool to know, because energy has to come from somewhere, right? Alright, this is just showing you everything. Cool. DNAP1. DNAP1's job is to replace the RNA primer with DNA complementary to the template. Just to let you know, DNA polymerase is not doing this out of thin air. There is a 3' end here, and I will explain how it gets there in just a moment. So, DNAP1 and 3, distinct functions. 1 replaces the primers, 3 adds to the primers. Okay? Cool. So, let's look at it all in like a short summary. Step 1, RNAP comes in, puts down the primers. Step 2, Step 2, DNAP3 comes to the 3' end of this RNA primer and adds DNA nucleotides. Step 3, DNA polymerase 1, DNAP1, comes and replaces the primers. And step 4, complete. Or is it? There is a problem at the replication fork. So, as we know, or as I just explained, DNAP3 can only add nucleotides to a 3' end. But we know that DNA is anti-parallel in nature. So if you have a replication fork here, and let's say you have an RNA primer over here. Here, let me write. So this would be 3', and that's perfect because that means that DNAP can just chug along this way. But if this is 5' here, then on this side, the lagging strand, 3' is over here. Did I get that right? Yeah, I think I got that right. Cool. So that means that there's a little problem because if it continues going this way, then it's gonna run out because helicase is going this way. The actual direction of replication is this way. So what's up? So what do we do about this? Here, this is another strand, or this is another diagram. At the replication fork, there's a leading and a lagging strand. So the leading strand is synthesized continuously. So that's this side here and this side here. But the lagging strand is synthesized in short, discontinuous fragments of about 1 to 2,000 nucleotides called Okazaki fragments. So these are Okazaki fragments, as you can see in the diagram. And Okazaki fragments are a little bit more complicated, and they have a couple more steps. Let's look at it this way. So what happens at Okazaki fragments? So everything can continue as like everything we learned before still applies. So a primer comes down, puts down a primer. DNAP3 extends it. DNAP1 replaces the primers. So basically DNAP1 would touch down here, not touch down here. For this example, DNAP1 would touch down here, go along, and stop right here, as you can see. And then there's the last step here, ligase has to come and join this gap. Oh. Ligase has to come and join this gap. Do you guys understand what's going on? Any questions yet? Alright. So DNAP1 and 3 can't actually can't actually ligate this band here. So because DNAP1 is coming down and replacing, it has to stop at this last base, right? And if it stops at this last base, there's still a little gap here because just because it puts a base down doesn't mean that it's ligating the gap between this base and this base. So basically, this entire series or this entire sequence of events has to happen at every single Okazaki fragment. So if I take you back over here, it'll happen here, it'll happen here, and then once the fork continues this way, it'll happen here again. Same thing here, here, and here. And it will happen here once for the leading strand because at the origin, the leading strand is on one side and the lagging strand is on the other side just because of the way that DNA works. Okay? And we're going to continue. What does ligase do? Ligase just creates a phosphodiester bond. So, pretty simple. Basically, this is the a little summary of the different machinery. All the machinery is pretty much the same, except for the fact that fragments have to be joined by ligase on the lagging strand. Unwinding happens the same. Cool. Let me show you guys a little video. So in real life, it looks pretty complicated, and there's a little diagram. It's more comprehensive than all the other ones. And B is basically showing how the fragments are like attached again. Here, I know this doesn't really make sense right now, so let me show you guys a little video. I don't think you guys can hear sound, so I'm gonna switch the screen that I am sharing. Can you guys see the Chrome page?
You're not streaming right now.
Oh, I'm not streaming right now? Give me a second. Is this working?
Not really.
Oh, what's going on?
It's just Chris a troll.
Wait, that's crazy. Give me a second. Oh, what's going on? Wait, give me a second, guys. I'm gonna reopen this. Yeah, here we go. Does that work? You guys can see it now? Hello? Hello?
Yeah,
We can see it.
Okay,
We
cool.
can see it.
Alright, I'm gonna play the video. Basically, this is what DNALC.org have created. It's like a model of DNA.
Based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an
Can you raise the
assembly
volume a
line
bit?
of amazing
Yep.
miniature biochemical machines that are pulling apart the DNA double helix.
Here, let me erase the video. Volume mixer. What was my
And cranking out
Can you guys hear it?
of each strand.
Barely.
The DNA to be copied enters the production
Here.
line from bottom left.
Can't
The
you just send the
whirling
link in like
molecular
the
machine
channel, and then we can watch it later on our own time
That's a good idea.
with like
Is
the
it
volume.
the same one as the one in the slides?
Yeah, it's in PowerPoint.
I think this one is not in the PowerPoint. This one is on our website.
Okay.
Okay.
Can you send it in the bio chat?
Yeah, I did.
Okay, thank you.
Let's like just look over it for a second, even if you can't hear it.
DNA as fast as a jet engine as it unwinds the double helix into two
So one thing to just note here is that in all the other diagrams, it looked like the enzymes were moving, but in reality, the enzymes don't move. The enzymes are anchored, and the enzymes pull the DNA through.
One strand
Oh, I
is
see.
copied continuously
Yeah.
and can be seen.
Is this like a real like is this like actually DNA or is this like a
Things are not
simulation?
so
This is a
simple for the other
simulation,
strand, because
but
it must be
it's
copied
pretty
backwards.
cool.
It is drawn out repeatedly
So the bottom side would be the leading strand, and then the top would be the lagging
in
strand.
loops and copied one section at a time.
So every
Wait, that's so
time
weird.
The
Like,
end result is two new DNA molecules.
Yeah. Did you have something to say?
oh wait, no, that makes sense because when it reaches the DNA, does the enzyme like fall off and then reattach?
Oh, here?
DNA ligase.
It spins the
Oh.
DNA as fast as a jet
Let's
engine.
go here. So, you see here, like, it breaks off.
As it unwinds the double
Yeah.
helix into two strands.
Right here, ligase pretty much like is part of this little complex, and then ligates it, and then just breaks off.
Okay.
And can be seen spooling off to the right.
So is there just another one floating
Things are not so
nearby?
simple for the other strand,
I think so.
because it
Honestly,
must
I'm
be
not
copied
entirely sure how it
backwards.
works, but I know that like that's pretty accurate,
It is
I
drawn
guess.
out repeatedly in loops and copied one section
Okay.
at a time.
Yeah, I guess they float
The
around.
end result-
Honestly, I can't really tell you how it happens exactly, but it must work. And I don't think you have to know it into that detail just yet. Right.
Yeah.
Okay, termination. So, DNA replication ends in eukaryotes
Wait,
when
can you start the stream again?
Oh,
Yeah.
yeah, you're right. Sorry. Let's go back here. Cool. So, termination. DNA replication termination ends when you either reach the end of the chromosome or two replication bubbles or forks meet. Uh, this is what it would look like in eukaryotes with linear DNA, and this is what it would look like with circular DNA. Give me 1 second, another person asks to be invited. Uh, all right.
When two bubbles meet, do the replicated strands need to be joined with ligase? Or
Yes.
okay, just making sure.
Okay. Let's see if there are some homework questions. I think you guys should do the homework questions. If you have any questions, feel free to ask Miss Law or me if she's not responding. Okay, telomeres. There is a problem at the ends of DNA, and it relates to aging. We think it relates to aging, and an enzyme called telomerase. So, does anybody know what telomeres are already?
Is it just the I'm not sure, but is it just the DNA at the end of chromosomes?
Yeah, so it's actually a specific sequence that's repeated. So,
Oh, I see.
it's repeated at the ends of DNA, and in normal cells, so in somatic cells, every single time DNA replication happens, a part of the telomere is lost. So they're kind of caps, they're like protective caps almost. So let me tell you why a part of DNA is lost. Let's look at one part or one end of linear DNA. So this doesn't apply to plasmids, which is circular. This applies only to linear DNA. DNA gets progressively shorter with each round of replication because
Battery low.
there's always going to be a 5 prime end here that can't be copied. Let's take a look.
Oh, I see.
Over here, the template splits, so 1, 2. The bottom strand is fine because it has a 3 prime end on the complementary to basically continue all the way to the end. DNAP can continue all the way to the end. Over here, where RNAP would have put down primers, and then for every other situation, there would have been, because there would have been an ORI here, and there would have been another strand that was continuing this way. Over here, there is no 3 prime end. So DNAP1 can't come and fill this gap. So what happens? This gap can't be filled. So we just lose information? No. So what we have are something called telomeres. They're non-coding and they consist of multiple repeats of a short sequence. And they're found at the ends of eukaryotic chromosomes.
Wait, so
Here's,
does that mean that when replicating one strand just stays shorter than the other strand?
yeah.
Okay.
One strand is just shorter.
Okay.
Yeah, and there's not much you can really do about it. But you should be fine, right? Like, you and I, we don't have problems. Uh, so what do telomeres do? They protect chromosomes from being eroded through multiple rounds of DNA replication because telomeres don't code for anything. So their existence protects actually coding DNA, if that makes sense. They're like a protective cap. Uncapping is actually sensed by cells, and we know that it leads to cellular aging where cells stop growing and dividing, which is called senescence, or programmed self-destruction, or apoptosis. So, at conception, cells have telomeres that are about 15,000 bases long. At birth, the telomeres are about 10,000 bases, And humans die not because they have a telomere 5,000 bases long, but just on average when humans die, telomere length is about 5,000 bases. So, as Ms. Law has added here, you're born with the longest telomeres you'll ever have. It only gets shorter with age. So let's look at a little thingy, little graph here, telomeres and aging. Yeah. So, a little question here. When would telomeres, oh, there's the answer here already, no. When are telomeres added to the chromosome? And yeah, when are telomeres added to the chromosome? Don't look. No. No. uh okay, so yeah, they're added prior to birth, prior to conception, and prior to fertilization. Oh, I guess that's not the full answer. What do you guys think? When do you guys think telomeres are added to chromosomes? Any guesses?
If it's prior to fertilization, wouldn't that be like during meiosis or something?
Okay, so for yeah.
Would it just be during replication?
So I'm looking for something a little bit more, maybe type of cell, you know?
Starts with a G.
Gamete.
Okay, okay. That is the answer. And another answer I could have accepted is like germline cells. So you're right. Meiosis. And cell types would be gametes and germline cells. Basically, same thing. They're also seen in cancer cells, actually. So there's a specific enzyme, actually, that adds your telomeres. And that enzyme is called telomerase. Telomerase is a ribonucleoprotein that extends your chromosomes using reverse transcriptase. I think you guys should be familiar with the term reverse transcriptase. And
No.
okay, that's fine. We'll go through it. Ribonucleoprotein. It's a it's a long word, but basically like ribo, like the sugar, nucleo, like ribonucleo, because that's like RNA protein. It's an RNA protein. That's what it means. And what it does is telomerase has its own like built-in sequence. Like it has a baked-in RNA sequence. And what it does is what this diagram is basically showing. I'll go through it step by step. Telomerase binds to the 3' flanking end of a telomere that is complementary to telomerase RNA. So as I mentioned earlier, telomeres are a repeating set of sequences. And that's because telomerase has its own unique RNA sequence. Does that make sense? So human telomeres have a different sequence from like different eukaryotes telomeres. So human telomerase has its own unique sequence. So what it does is it comes here, a section of it, or whatever, a section of it matches. So it comes and attaches to the 3' end because the 5' end is the problem. But right now it's attaching to the 3' end. And what it does is using this RNA sequence, it extends this 3' end. Boom. Once the 3' end is extended sufficiently, RNAP can come in again, prime, and then DNAP can basically fill in. Does that make sense?
Mm.
The reason why this couldn't happen before is because you didn't have enough space. But now you have this super long 3' end, or this super long template strand, so you have more space for a primer to come in, DNAP 3 to come in, and then DNAP 1 can come in again, and then you can basically extend your telomere to prevent losing actual coding DNA down the line. Yeah, it's pretty simple. It's really, really important, though, to understand where the telomeres would be. So let's say that I gave you like a fork. I gave you a replication fork like this, and you had to label like 5', or that's not even a 5. Like you had to label 3' and 5'. You would have to be able to tell me where the telomeres would be. And if I'm asking telomere, I'm asking for this gap. Does that make sense, guys? You have to be able to understand at a replication fork which side or which end of which DNA here would have a problem with telomeres, if that makes sense. So in this example, if this is 3' and this is 5', the problem would be at this end. Wait, give me a second. No, I think we'll cover that in just a little bit further. All right.
Wait, I have a question.
Yeah.
For the slide before,
Mhm.
it says telomerase binds to the end of the telomere, right?
Mhm.
But where does the telomere form in the first place?
As in, how does this like exist in the first
Yeah,
place?
how does that exist for the telomerase to continue it?
Like your parents.
Oh, so it's just built off of their existing telomere?
Yeah, because DNA would have had to come from your your ancestors or your previous family.
Oh, okay, okay.
So you would not really, yeah, you don't start off from like nothing. You inherit your the DNA from your parents. You inherit telomerase from your parents.
I see, I see.
So yeah, that's the cool thing about life. Like you don't know where it really started, but it works this way. So yay. Are there any questions? This is a little bit of a more concise diagram about how telomerase works. If you don't really understand it yet, it's fine. You'll have a little bit of time to digest this information, come back to it. I know that I didn't really get this the first time I saw it. I had to think about it a little bit longer and then it made more sense to me. So if you look over here, if this is the existing, the dark blue is the existing DNA. Telomerase comes, adds light blue. Pink is its RNA strand that's like native to itself. So this extended light blue is coming from telomerase, but this light blue section is not coming from telomerase. It's coming from all the machinery that we already know about. And all the machinery that we already know about can work here because of telomerase's job extending this 3' side. Reverse transcriptase is an enzyme that can synthesize complementary DNA from an RNA template. Does that make sense? So it's called reverse transcriptase because normal transcription turns turns DNA instructions, or I'll just write D, to RNA, but it's going in reverse. So that's why we call it reverse transcriptase, because this process of turning DNA information into RNA, into protein, so this is called transcription, but we're going backwards, which is why we call it reverse transcriptase, an enzyme that can do that. So yeah, telomerase is only found in certain cells. It's only found in germline cells and cancer cells. Cells that have telomerase live for a longer period of time because, as we talked about earlier, the uncapping of cells is related to senescence, and senescence is related to basically cells ending themselves. The lack of telomerase in most cells can explain why cells have a finite lifespan, um, but I don't think that there's definite research just yet. So, what would happen if germline cells didn't have telomerase?
You would lose part of the genetic information.
Yeah, so let's say you had a son or a daughter and you have no telomerase. If you
They just wouldn't be
started,
able to grow.
so what would
They just wouldn't be
happen?
able to grow up. They would die because they don't have enough of telomeres at the end of their chromosomes.
So what happens if you
I
don't
guess
have
you would,
telomeres?
I guess you would lose that
The cells
extra
die
overhang.
Pardon, Carolyn?
faster. Cells die faster.
So why would you die though? What's important here? What are telomeres protecting?
They protect the actual cell information, but then didn't you say that cells can like detect when there's no cap?
Uncapping. Yeah,
So yeah, so then after that the cells would like uh go undergo cell death,
yeah.
which means cells would die.
Yep, that's the answer. So there's a couple videos here for the reading. Some homework questions. Oh, I don't think you can answer this just yet. We haven't covered mutations and repair, but you might be able to answer the other ones. Cool. This is a short recap of all the different terms, all the different machinery that we learned about today. Yeah, any final questions?
Yeah, I have a question.
Yeah, go ahead.
Okay, so this is going a little back, but when we went to the start of telomeres.
Okay, give me 1 second.
So you know how
what slide?
said that, yeah, you said that there's the gap. Well, yeah, the one right after.
This one?
Right after that. Wait. No.
The one before it?
It's the one with the no, yeah, wait, yeah, that one. Yes, the one right after this slide. Yeah, that one. So you know how there's the gap that's unfilled?
Mm-hmm.
So you said that it's because of the RNA primer that was there. So why can't the DNA whatever it was come and replace it?
Okay. Yeah, that's a really good question. So we know that all the DNAPs, so DNAP1 or DNAP3, in this case, it would be DNAP1's job to remove this primer and continue the strand, right?
Yeah.
But the reason why it remains unfilled is because DNAP1 still needs a 3' end to build off of.
Okay.
Let's take a quick look at this slide. This slide explains it a little bit better. So let's assume that this light blue section was not here. And the primer was actually here. If DNAP1 needs a 3' end to add DNA bases here, is there a 3' end here for DNAP1 to add off of?
No.
No. So let's look at the situation where telomerase does its job. This light blue section exists, so the primer is actually here. Essentially, what's going on is the problem section is just being pushed further right. The problem of DNAP1 not having a 3' end is always going to exist, and there's always going to be a gap, but essentially it lets you go through one more iteration of replication before this gap. So when telomerase does its job, the primer is here instead of here. So the primer is here, DNAP3 comes in, and DNAP1. Okay, that doesn't really answer your question though, does it?
Would it start from the left side of the rectangle because that's the 3' end?
Mm-hmm. So the 3' end is here, but there is a 3' end. Let's take a look. I understand your question, and that doesn't really answer it. So let's take a look at this section. During normal replication, when you have a primer, DNAP3 is going to stop where this primer begins. Then DNAP1 will come and use the 3' end placed by DNAP3. So normally there would be a 3' end here, and that's where DNAP1 comes in and replaces the primers. Does that make sense?
Okay.
So since DNAP3 was able to do its job, like further down, there's a 3' end for DNAP1 to come and replace the primers. At the end of linear DNA, there is no DNAP3 that has done its job. Does that make sense?
Okay.
So the 3' end is here. There is no 3' end here. And that's why there's a gap.
But how does the primer get removed? If you scroll down just a bit.
the primer is removed.
Yeah.
To be honest with you, I can't say that I know the actual mechanism. I don't know if DNAP1 can remove primers but not add DNA bases if there's no 3' end. Does that make sense? Because DNAP1 is doing two things. It's removing the primer and then adding a DNA base. So
Yeah.
my best guess would be that it can still remove the primer, but it can't add a new DNA base to replace it.
All right. All right.
And that's a really good question. Maybe you can ask Miss Law, or I'll try and look it up. What happens to
Wait,
the primer?
Alex.
Yep.
That's fine. Quick question. So on the exact slide that you're on, for the very top picture, so you know how it shows that yellow box that it's a telomere, right? So does that mean the telomere for the lagging strand is shorter than the telomere for the leading strand, and when you do the whole telomerase thing, it creates more telomeres?
So,
or am I just thinking about this all wrong?
I understand your question. So the funny thing is let's say that we have an ORI here. We have an origin of replication right here. The problem here is that if you have a leading strand over here, which that's not true. This is a lagging strand. This is the top side. Then this side is a leading strand. So this is a lagging strand and this is a leading strand. They're not showing it, but over here there would be a gap too.
So they're actually the same size.
So if you really look at it, they're kind of the same size. They can't be like translated, but any problem that happens here happens here. Does that make sense?
Oh, so wait, does that mean that you're not losing DNA then? You're losing telomere length?
Yes.
Oh.
This entire box that we call the telomere is a repeating strand. It's non-coding, it's repeating, only exists to protect the DNA, and it's on here, and it's on this side too.
Oh, so that's why it becomes shorter when you age, because your cell replicates more?
Yes.
So you just lose more each time. Okay, okay.
Yep. So you see how there's a gap here, right? The next time you do replication, like this section will be missing, and then you'll have another gap here. If that makes sense.
Kind of.
Yes, there's a gap on both sides of all DNA strands after replication. Because any problem that exists on this side, the anti-parallel nature of DNA dictates that there will be another 5' strand here that has the same problem as the 5' strand over here.
Okay.
Wait, but the 3' strand, it stays the same length, but the 5' strand just gets shorter and shorter? Is that all? Like the gap just gets bigger and bigger?
So the gap doesn't get bigger and bigger because let's take a look at a situation where, let's look at the next replication from this point. If this is taken as the template strand, right? And what happens essentially is that its telomere, its telomere on this side is going to get shorter. Okay? And then let's take the bottom strand as the template. Then the telomere on this side will just get shorter too. Does that make sense?
I think so.
You're losing information on both sides. Each strand will become a template for another strand. We say template and complementary right now because we're looking at two strands, but we have to look that each complementary will then be a template for another strand in the next replication. So don't get your head mixed in too much about it. Just know that at the 5' end of linear DNA, you will have a gap. And there is a 5' gap on both sides.
All right, all right.
Is anybody still really confused? Maybe I can find a video if you guys are still really confused by tomorrow.
Wait, Alex.
Mm-hmm.
So you know how you were talking about the origin right there, right?
Yep.
It's like what happens at the origin? Because it's a leading strand, and then suddenly on the other side of the strand, it's a lagging strand. It like doesn't continue. It's like the origin like a wall.
So let's take a look over here. This slide can help you. Let's take a look at this slide. Okay. So we place an origin right here. To the left of this origin on the top side, so we'll call the top template and bottom complementary. So template strand. To the left, you have a 5' to begin, which means you have perfect, you have a leading strand. But the fork, there are 2 forks for every origin.
Okay.
1 second.
Mic off. Mic on.
Sorry. So you have 2 sorry, is it even, yeah. You have 2 forks for every origin. Leading and lagging is in relation to the fork.
Okay.
So if you have 2 forks per origin, then that means you have 2 leading strands and 2 lagging strands per origin.
Okay. Yeah.
It's all based on like perspective here. If you have a 5' here, then you have a 3' here.
Yeah.
But maybe a better way to look at it is at the fork. If you have a 3' here, then you have a 5' here. So you have a leading strand and a lagging strand. On the other side of the origin, when another fork is there, you have like an opposite leading and lagging.
Okay.
Does that make sense?
Makes sense. Yeah.
And this is why maybe like this understanding is helpful to understand the fact that telomeres exist on both sides. Because whenever you have an origin, it's like mirror. Does that make sense? Any other questions?
Is there something on the DNA that decides where the origins are going to be, or is it just the origins appear randomly?
Oh, that's a very, very good question. In your textbook, there may be a better answer, but origins of replication have specific locations.
Or what determines them?
So for DNA replication specifically, I'm not entirely sure exactly what those like factors are, but I know that for replication to begin, there needs to be a lot of different proteins and a lot of different things have to like come together, because remember that it's a complex that does this replication. So I'm guessing it's where the complex can attach properly. And obviously there will be multiple points of attachment. And for a plasmid, I'm not too sure, but they call them origins of replication. So my best guess would be that there must be something going on. That I could probably look up for you too.
So is it like a specific sequence of base pairs? Because there's not really anything else that can distinguish parts of DNA.
That's a good question. That's a good hypothesis. I will try and give you an answer. Maybe you can ask Miss Law. She might have an answer. I'll try and ask her too. That's a good question. I'll try and give you an answer for tomorrow. I don't have one for you right now.
Okay.
Any other questions?
I just really hate telomeres. Anyways, for basically you're saying the lagging strand is shorter because it can't go all the way, right? But doesn't that mean you're still technically losing DNA because you're just when you're at the end for the telomerase, it's just creating more telomerase, right? So aren't you still losing DNA or?
So
because at the end, this thing when it's helping, you're creating the sequence you're creating is more. You're not creating more like DNA that you had. It's telomerase.
So basically, you can think of here. Let's say that you have like kneecaps, okay? And you are going to fall 10 times a day, okay?
Love this.
No matter what, you're going to fall 10 times a day. Okay, and you're not just like falling onto like smooth ground, like this is like rough concrete we're talking about. Every time you fall, your kneecaps will be damaged.
Okay.
If you don't regularly like protective pads, okay? Let's say if you don't regularly maintain your protective kneecaps, eventually your knees will suffer.
Okay.
Your knees hidden behind your protective kneecaps are all the coding DNA that must be protected. Your protective kneecaps are the telomerase, is the telomere sequence. Telomeres do not code for anything. They are dispensable, but they have a very important purpose. Okay? So during your lifetime, this protective like kneecap is not going to deteriorate up to a point where you like start losing DNA, like normally. But they do have to be repaired. So in your germline cells, when you pass down your protective kneecaps to your offspring, you want to give them brand spanking new ones so that they can continue to fall 10 times a day throughout their lifetime. This is what the
Lovely. Okay.
telomere is. So yes, you lose. You do lose like part of the telomere sequence anyway. But the telomerase doesn't care. It just has to be long enough. Does that make sense? It just
Okay.
needs to have
Okay.
enough
Okay.
filler. And that's what
Okay.
it's doing. So obviously by the end there's still a gap, right? But telomerase
Yeah.
like that doesn't care because it's still effectively added this much.
Okay.
It has still effectively added this much.
Okay. Okay.
Yeah. Any other questions? And yeah, if you have questions like later in the day when you're studying, ask Ms. Law or put them in the bio chat and then I'm sure they can be answered eventually. So yeah, I think if that's all the questions, that's it for today, I guess. Thank you for coming in. Good luck studying. I know this is
Thank
a
you,
lot to
Alex.
digest. I'll take more questions tomorrow at the start if you have any. So
It was really helpful.
yeah, no
Yeah.
problem.
Thank you.
Yeah, thanks, Alex.
Thank you.
Thanks, Alex.
Thank you.
Thank you.
You're welcome.